Hello everyone and welcome to today's webinar, utilizing the Gibco CTS Rotea System, a versatile closed manufacturing system for cell and gene therapy development. I am Jacqueline and I'll be your moderator for today's event, and I welcome all of you to this event. Today's educational web seminar is presented by LabRoots and brought to you by Gibco by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. As we are addressing the Asia Pacific region, we have invited our audience to type questions in English, Mandarin, Korean, or Japanese. Our panelists will answer as many questions as possible at the end of the presentation. I would now like to welcome our three speakers, Dr. Premkumar, Cell and Gene Therapy Regional Field Application Scientist at Thermo Fisher Scientific, Emily Benz, Process Development Engineer at Biosite, and our third speaker, Eric Dubofsky, Biomedical Engineering Master Student at Cal Poly San Luis Obispo. Prem will now introduce the Rotea and its various applications for close processing of cell and gene therapies.
Thank you so much, and I would like to welcome you all for this webinar. Now, let's look at some of the main issues in cell therapy manufacturing. So you can see on the left a typical autologous T-cell therapy workflow. As you may already know, it is a very complex and labor-intensive process which requires many open manipulations, making it prone to errors and contamination. In addition, how to synchronize different instruments and processes, making the workflow trackable and compliant with regulatory requirement, which is another layer of complexity. So based on these issues, we understand customers are looking for three main solutions: closed system to close the process, modular system to remain flexible, and automation to reduce labor and human errors. Okay, so this is rather an important slide. I would like to spend some time explaining the benefits of closed automated autologous CAR-T options compared to that of a manual processing. So you can see that option one is the manual process. You need a large and expensive facility with dedicated Class B processing areas. So it is labor intensive and it's prone to contamination. And option two is the closed automated system with integrated incubation. We can do parallel processing in Class C processing area with minimal labor, but the processing instrument is poorly utilized. This is mainly because the incubation process is integrated into the closed automated system. To explain in simple terms, within a typical CAR-T process, we would expect at least 7 to 14 days of incubation, during which the entire instrument is tied up to one patient sample, which means if you want to process 2,000 patients per year for a 7-day incubation period, you would require at least 80 instruments, which is a very expensive things to set up. On the other hand, option three is the closed automation system using modular instruments with centralized incubation in single-use vessels such as static cell culture bags or G-Regs or Rocker bioreactors. This is more of an appropriate and a cost-effective strategy, meaning instead of having all the processes such as cell separation, incubation, formulation integrated into a single instrument, you would have modular instruments to carry out each unit operation of the workflow. And this is what Thermo Fisher is currently providing. Okay, so this is a vision for our cell therapy manufacturing workflow. We provide best-in-class closed modular and automated instruments in combination with superior reagents, protocols, and analytical solutions. We are taking a modular approach in closed automated cell therapy manufacturing with a centralized incubation. The graph below depicts our approach for an end-to-end T-cell therapy manufacturing platform. So starting from Rotea, which I'll be talking in detail later, PBMCs are isolated and directly transferred into our CTS Dynamax system for magnetic T-cell isolation with our CD3, CD28 Dynabeads. Once the T-cells are isolated, they can be transferred using the G3 lab controller into an incubation system of choice. Here we have shown our high-performer bioreactor. And after the T-cells are expanded, the entire process just goes through the reverse sequence. The expanded cells are transferred back into the CTS Dynamax to remove the residual Dynabeads, and the cell suspension is washed and formulated for cryopreservation using Rotea again. So while we can physically integrate these instruments, we are also enabling digital integration platforms, and our goal is to provide direct connection to Delta V platform to support full automation. Overall, we can promise a full integrated workflow with a scalable and robust manufacturing performance. Again, shown here is the schematic of a gene-modified T-cell therapy workflow, and we have products that support every step of the workflow. So for selection of T-cells, we have our CTS Dynabeads and Dynamag magnet for large-scale T-cell isolation. And for viral-based gene modifications, we have a variety of tools such as the CTS LVMAX and a new AAVMAX prototype. And for non-viral based methods, we provide CTS TrueCut Cas9 protein and talents, which can be delivered to cells with our new large-scale CTS electroporation system, which is scheduled to launch this year. And after gene modification for T-cell expansion, we have a variety of serum-free media choices, such as our CTS Optimizer, specifically designed for autologous T-cell expansion, and our CTS Optimizer Pro for allogenic T-cell expansion. And finally, our CTS AIM5 media for expanding a variety of other immune cells. And now I would like to introduce our recently launched closed and automated solution called CTS Rotea for cell and gene therapy, a bean-to-bean process. So you can see that from starting from the PBMC isolation from a leukopak and throughout the process, there are multiple steps of cell wash, concentrate, media or buffer exchange, and cell separation as well. So Rotea can basically do it all, but most importantly in a modular way and centralized incubation. Okay, so Rotea is a compact, closed, automated cell processing system designed specifically for cell therapy manufacturing, and I would like to highlight 4 key benefits here. So with proprietary counterflow centrifugation technology, it allows customers to perform flexible operations such as cell separation based on size, wash, concentrate, buffer exchange, and formulate. The unique chamber design allows very low output volume of 5 mL with superior cell recovery and viability, which is typically more than 95%. So no other product in the market can deliver or achieve a low output volume of 5 mL, which is very critical to obtain high cell density concentration as an output. And this is required for operations such as electroporation and formulation. And further, the most important point, the OPC UA interface supports 21 CFR Part 11 compliance. We will also provide extensive regulatory support documentation for the instrument and the single-use kit. Okay, now let's look at a closer look of the whole system. The compact system allows easy integration into your process. It's only 20 kgs. The closed single-use kit is sterilized by gamma irradiation and has flexible input and output ports, which allow fast and multi-process integration. We have 2 types of kits, the standard kit with a flow rate of 5 to 110 mL per minute, and for processing larger volumes, we have a high flow kit with the flow rates to 30 to 160 mL per minute. And on the software itself, with the unique CellCam technology, you can visualize your cells while you're processing it. And this is the unique feature of Rotea, which you cannot see in other competitive products in the market. And you can adjust the G-force and the flow rate settings during the process development. So the user programmable GUI allows you to easily create your own protocols, but however, we provide a preset protocols for different applications. Now, let's look at how the counterflow centrifugation works. So you can see here, there are 2 forces, G-force and the flow rate, acting in opposite direction. So first, cells enter the chamber through a narrow tube at a certain flow rate, shown in red arrow, and the high fluid entry velocity ensures that the cells exit the tip of the chamber. At the same time, the chamber spins at an opposite G-force to prevent cells from exiting the chamber. So when the flow rate and the G-force are balanced, a fluidized bed of cells are formed in the chamber and it's maintained in the chamber. So you can see that when in a normal manual centrifugation, what you form is a pellet. Here the cells are happily floating in the buffer. So at this state, you can perform media exchange, cell wash, cell separation by size, and also concentrate and harvest. And due to the conical shape design of the chamber and the narrowing of the chamber, which increases the fluid velocity and forces some of the smaller cells out through the top of the chamber first. So this process is called elutration, how you do this is basically you reduce the G-force and increase the flow rate to separate the smaller cells from the larger cells. Instead of elucidating of just the smaller cells, you could also choose to deplete unwanted smaller cells. For example, when you're loading a leukopak, tiny platelets and plasma immediately leave the chamber while you're loading it, and the PBMCs are retained in the chamber. You can achieve platelet depletion simply from sample loading again, and in fact, Rotea is able to deplete debris and small-sized dead cells, which gives its consistent achievement of high cell viability. And as I mentioned earlier, the G-force in the flow rate settings can be adjusted to separate smaller cells. Again, the G-force is lowered, allowing the smaller cells to leave the chamber and collected in an intermediate bag. So this is an example of how we perform monocyte and lymphocyte separation. So you can see the lymphocytes are shown as the yellow beads, which are 6 to 7 microns, while the monocytes are shown as the blue beads, which is an average of around 10 microns. So to achieve higher purity of larger cells or the monocytes, we can choose to optimize the volume of elucidation buffer to collect the small cells, such as the lymphocytes. So next, you can concentrate and harvest the monocytes into a separate bag. Further, you can bring back the elucidated lymphocytes into the chamber for volume reduction and harvesting separately. So in line with this, we have evaluated Rotea for processing different cell types such as PBMCs, T cells, MSCs, Act293 cells, IPSC Ferroids, and there are limitless opportunities to perform a variety of applications such as cell isolation, small to medium scale processing, cryopreserved cells, wash media exchange, formulation for cryopreservation, RBC lysis or depletion, LV or AAV clarification, and the list keeps growing. So to put in simple terms, it's a single instrument, a single kit, but multiple applications. And more importantly, it's a modular system. So once one process is completed, the system is basically free for the next step. In this study here, a single donor leukopak was split into two, and we wanted to compare the efficiency of PBMC isolation using manual FICOL protocol and the closed automated Rotea separation with no FICOL involved. So you can see that we can also do, the key takeaway point here is the Rotea can isolate PBMCs in a closed process within less than 30 minutes with equivalent performance to FICOL. There's a common question in the field that whether RBC lysis process impacts the viability of the PBMCs and quality of the cell composition. So you can clearly see from the data here that the RBC lysis process in Rotea does not have any impact on the cell population, with the recovery and the viability being high. And we also have protocols now to process other samples such as the whole blood and Buffy code using hetastarch or any RBC lysis buffer. Next, after PBMC isolation, using RBC lysis in Rotea, we could separate the lymphocytes and monocytes fraction by just optimizing the G-force and flow rate settings again. So in this demo, we took a fresh leukopak sample, carried out an in-chamber lysis followed by monocytes and lymphocyte separation. So before RBC lysis, the fresh leukopak sample had total of 3 billion white blood cells as an input with 22% monocytes and 55% lymphocytes. After we did the RBC lysis, the total PBMCs were more than 90%, and we managed to separate more than 67% of the monocyte fraction in the monocyte harvest bag. On a separate note, the lymphocyte fraction has more than 80% enrichment, but the data is not shown here. So depending on the starting donor PBMC composition, the separation efficiency will be varied, but we can adjust the G-force and the flow rate settings in the model feature to entirely optimize and maximize the enrichment of each of the cell populations. And again, here are some data showcasing Rotea's capability. As I mentioned, we can get more than 95% T cell recovery and viability after wash and concentrate, and this holds for other cell types as well. And you can see that the maximum number of T cells accumulated in the chamber, and the counterflow centrifugation chamber volume is 10 mils and can hold up to 5 billion T cells or 3 billion adult stem cells, but depending on the cell size and the density of the media. Although the chamber size is only 10 mils, continuous processing can be performed using Rotea with multiple discharges if you have more than 5 billion cells to process. And like I mentioned, the output volume can be as low as 5 mils without compromising the recovery and the viability. This is a great feature again for preparing for your electroporation or formulation. And at last, the T cell quality and ratio is also not affected by the Rotea process. So similar to the T cells, we have washed and concentrated freshly expanded NK cells using our new proprietary feeder-free NK expansion prototype media. On the left, you can see around 650 million NK cells bed formed in the chamber. On the right, you can see that the pre and post Rotea viability did not shift and maintained around 97%, and the total cell recovery was around 84%. And the purity of the NK cells measured by the CD56 marker shows a similar range for pre and post Rotea. So it's kind of clearly evident that with highly sensitive cells like T cells and NK cells, the Rotea is very gentle and retains the population as well. And Rotea process is flexible and can fit into the MSC manufacturing workflow as well, either if you are in 2D monolayer or 3D microcarrier-based suspension culture. So starting from a working cell bank, Rotea can be used to thaw cryopreserved MSCs, wash media exchange with fresh media, and transfer into a cell factory or bioreactor directly for expansion. So for each step in the process, Rotea can be used for volume reduction, wash, concentrate the harvested cells, either for further processing or for formulation for cryopreservation. But for microcarrier cultures, we recommend Harvey stainer upstream of Rotea to separate the cells from the microcarrier. And yeah, you can see here that Rotea can be used for processing small to medium to large scale cultures up to 20 liters, but depending on the robustness of the cells. You can use Rotea to process cells by directly harvesting from single-use expansion systems. So for small-scale process optimization, we process cultures less than 500 mils. You can connect the syringes for easy evaluation of cell counts. You can also see that Rotea can be connected directly to cell factories or bioreactors, or you can transfer the cell suspension to a larger bag and you can process. So it's a very flexible instrument. You can connect both upstream and downstream systems. Okay, here we carried out an experiment for washing cryopreserved patient-derived MSCs with two different buffers. On the left, you can see 100 million cells in the chamber. Post-thaw viability was around 90%. And on the right, you can see Rotea performance in terms of recovery and viability with one or two wash steps. So compared to the manual centrifugation of only 50% recovery, Rotea managed to achieve an average of 75% recovery and also maintains more than 80% cell viability. The key point that I would like to highlight here is Rotea can be used to wash away impurities such as residual proteins, growth factors, chemicals from your spent media. So in the table below, with one-time dilution of cells and one-time wash, we could remove more than 200-fold of residual VSA. And with one-time dilution of cells and 2 times wash, we showed that we can deplete more than 3,600-fold washout efficiency. Okay, so similar to the T-cell and MSC application, Rotea is again flexible to incorporate for viral vector manufacturing as well. So here in this experiment, we did for LV clarification of suspension viral production cells. On the left, again, you can see around 300 million VPCs in the chamber, forming a nice fluidized bed, and the lentivirus containing supernatant is collected in a bag for further concentration and purification. So we managed to achieve 100% separation of X cells from the supernatant, and the titration flow data on the right, you can see that comparable or slightly better transduction efficiency than the manual centrifugation method. Okay, so moving further, this is another example to show Rotea's capability in processing spheroids or organoids or aggregates in a gentle fluidized bed without disrupting their morphology. As you can see that the IPSC Ferroids are around 440 to 200 microns in size that are captured in the chamber, while the smaller polymers around 20 microns are washed away from the chamber because of the counterflow centrifugation technology. And on the right, we carried out different G-force and flow rate settings to optimize the conditions for high cell recovery and viability. So you can see that 350 Gs and 40 mils per minute, which achieved more than 86% recovery with morphology being healthy and intact. Okay, talking about IPSC Ferroids, we recently launched our STEM-scale PSC suspension medium, which is a scalable, easy-to-use medium that supports robust expansion of pluripotent stem cells in suspension, delivering 5 to 10-fold expansion per passage. The medium uses a spheroid self-assembly method to promote PSC growth and does not require the use of microcarriers. Beyond expansion capability, the workflow of change medium and passage cells is fairly straightforward and therefore amenable to multiple culture formats and automation. The stem scale medium promotes consistent spheroid formation and expansion capability across multiple lines and maintains pluripotency across multiple passages. Now, back to the crux of this webinar, our collaborators from Biosite will share more about their experience using Rotea for IPSC spheroid processing. Over to you.
Thank you, Prem, for providing insights on the various applications of Rotea. I would now like to invite Emily from Biosite and Eric to share more about their work around the use of Rotea for processing pluripotent stem cells.
So Biosite is a regenerative medicine company developing stem cell-derived cell therapies that hold the promise of a functional cure for patients with type 1 diabetes. Our cell therapy is a combination product, and so it's comprised of stem cell-derived insulin-producing cells encapsulated in a immunoprotective device. And so after implant, the device becomes vascularized to allow for nutrient and small molecule transfer across the membrane. And so our insulin-producing cells can then respond to blood glucose levels. As you can see by the graph here, we have performed preclinical studies that show successful control of blood glucose levels. The x-axis is time in days, and the y-axis is showing blood glucose levels. The green line is a non-diabetic mouse, and so you can see stable levels. And then the blue line is a diabetic mouse where you can see extremely large fluctuations. However, after implant and over time as our cells mature in vivo, they begin to respond to glucose levels and can effectively control them, similar to that observed in the non-diabetic mouse. And so the overall goal is to provide long-term insulin independency to diabetic patients. So our production process occurs over about 30 days. Initially, we thaw human embryonic stem cells and expand those in flasks and cell stacks over a period of 15 days and to generate enough cell product to then aggregate and perform a directed differentiation down the pancreatic progenitor lineage. Upon the final day of differentiation, our pancreatic endoderm cells, or PECA1, are frozen down for long-term storage, and then upon need, they are thawed and seeded into one of our device configurations, where that device is then implanted into our patients. Worldwide, there is an increasing incidence of type 1 diabetes. So to support the millions of diabetic patients, we would require a commercial scale production of our cell manufacturing process. So to meet this demand, one option would be to scale out our process. Scaling out involves multiplying the number of batches. And so in our case, that would lead to hundreds of roller bottles. And as you can assume, that becomes unrealistic very quickly, as this leads to high aseptic risk, a large production footprint, and higher costs. Therefore, it's important to develop a scale-up model where we increase batch size to support large-scale production. So scaling up a cell therapy requires multiple considerations, a few of them being process comparability, so ensuring that across batches and across scales, you're producing the same product, you're maintaining product integrity. Additionally, it's important to establish a great relationship with your vendors and even consider multiple vendors for various consumables and reagents. And then additionally, being aware of the facility capacity needs and whether you perform your production process in-house or in collaboration with a CMO. However, the focus of today's presentation is really identifying scale-up technologies that meet the process requirements. So for our process, we have decided to transition to suspension bioreactor systems because they're closed, controlled, and scalable, which allows for a robust commercial production process. And so in conjunction with the bioreactor, we require a cell separation system to perform closed media exchanges and harvesting processes, which leads us to the Rotea, And so Eric will be discussing how we've incorporated the Rotea system as a suitable technology that meets our needs.
Thanks, Emily. So first, some details on the aggregates and the drug substance that we are using, that we're processing with the Rotea. Viasite cell drug substance, or PEC-01, consists of pancreatic progenitor aggregates. These aggregates are pretty dense. They're comprised of multiple cell populations at different phases of differentiation. There's also extracellular matrix in each aggregate. Usually we see aggregates between 200 and 400 microns in diameter. The aggregates typically grow in size as we move along the differentiation process. So, like Emily mentioned, we want to use the Rotea system as part of our aggregate production process. And the Rotea system is good for this application because it addresses several of our requirements for aggregate processing. Specifically, it allows us to accomplish media exchange, passage, and harvest of the aggregates and cells while maintaining a closed environment. It's also an efficient processing regime. The entire process should take less than an hour in total. There's no effect to the aggregate viability, morphology, or characterization after processing. Rotea is also a gentle processor, and its single use allows us to maintain a closed environment and minimize aseptic risk. It also allows the isolation of a specific cell population, so we can separate the aggregates from non-aggregates and also from any dead or single cells that we don't want in our final harvest population. And then finally, as Emily mentioned, it is a scalable technology, meaning that as we scale up our production process, the Rotea will be able to be scaled with us. So a brief overview of the PEC-01 scale-up development workflow and where the Rotea fits into that workflow. To start our production process, we expand human embryonic stem cells from thaw into 2D adherent culture on flasks, move from flasks to cell stacks, and then we aggregate our cells in preparation for differentiation into the final cell product. This differentiation process occurs in suspension bioreactors, and this is where we see the Rotea being integrated into our process, allowing us to have a closed process media exchange and harvest, and also efficient and streamlined processing to process our aggregates and remove any non-aggregate debris. After this part of the process, we then move to cryopreservation and post-thaw culture, which finishes out the production of our cell therapy product. So in order to integrate the Rotea into our process, we first had to perform some initial testing with our aggregates just to see what parameters would be good to maintain aggregate morphology, integrity, and viability as they were processed. And the way we did this was with just a very basic initial testing setup using a couple of bottle assemblies I'm going to walk you through here. And this allowed us to see what flow rates and what centrifuge speeds were best suited for maintaining aggregate morphology, integrity, and viability. So first, we drew aggregate cells in through an input bottle. This would be representative of our suspension bioreactor. The cells were drawn into the Rotea counterflow centrifugation cone, and we tried to form beds at various flow rates and centrifuge speeds. The spent media from the input is sent to the waste along with any non-aggregate cell debris that was removed from the fluidized bed by the Rotea. The aggregates still in this counterflow centrifugation cone were then washed with fresh media. And finally, the processed and concentrated aggregates were sent from the Rotea to a harvest bottle where we could then confirm that the aggregates were still viable and still intact. And this setup allowed us to narrow down the range of flow rates and centrifuge speeds that best maintain our aggregate properties that we were looking for. So like I mentioned, there were two process parameters that we were primarily interested in optimizing. One is the pump flow rate, and the reason we're interested in this parameter is because if it's too low, we have a slow processing time with the Rotea, but there may be some improved aggregate retention. Conversely, if pump flow rate is too high, we get faster processing, but we might have lower viability or retention of our aggregates. On the flip side, when we're optimizing centrifuge speed, if the centrifuge speed is too low, you won't retain any of your aggregates. They'll be eluted directly to the waste. Conversely there, if your centrifuge speed is too high, you're going to have low aggregate viability, and you might also retain some of the debris that you don't want in your final product. So these were the two primary parameters that we were interested in optimizing using the test setup that we just saw. It is worth mentioning there are several other parameters that may affect the process inputs or outputs. However, these were assumed constant for testing for practical reasons. Some of these include the media density and viscosity, the density of the aggregates themselves, the diameter of the aggregates, and the number of wash steps performed by the Rotea during our process. So one of the other things that we were evaluating during these initial tests was we wanted to make sure that our process actually formed a fluidized bed in the counterflow centrifugation cone. And this is important because the fluidized bed maintains the integrity of the aggregates and also ensures that they are correctly retained and sent to the harvest as opposed to being sent to the waste. And as you can see on the picture on the left there, we were able to successfully form a nice looking fluidized bed in the counterflow centrifuge cone. We're also interested in evaluating the viability of the aggregates after processing, and this could be visually seen by any turbidity in the counterflow centrifuge cone during processing. And also post-process, we would look at the aggregates under a light microscope as seen in the middle picture there and confirm that the aggregates were intact, and then using software to make sure that our diameter after processing is comparable to the diameter before processing. We would also look at the viability before and after processing as seen on the graph on the far right. Pre-test, we would see aggregate viability in the high 90%. Post-test, we would also see comparable viability after Rotea processing. Another thing we're interested in for our production process is processing the aggregates over multiple days over multiple media exchanges. And this is because, as Emily mentioned, our production process takes several days. So we want to be able to use the Rotea for multiple days if possible without any negative effects on our aggregates. And so during our testing, we processed the aggregates for multiple successive days in a row and observed a minimum loss in viability, as seen on the graph on the left here. Over 3 days, we would process the aggregates once each day with the Rotea, and we would see minimum change in viability after each processing, at only about 3% loss in viability after each test. We also looked at diameter after multiple successive processing days, and once again saw a minimal change in aggregate diameter, as seen on the graph on the right. In fact, on day 2, we actually saw a slight, not statistically significant, but a slight increase in aggregate diameter, suggesting that we're in fact removing some of the non-aggregate debris that would have small diameters. So that's a good thing. We wanted to see that debris get sent to the waste instead of the harvest. So now that we've established the approximate initial conditions that we want to see integrated into our production process, we can also look into possible future applications of the Rotea to our cell therapy production process. So this is our overview of our production process: expansion, differentiation, cryopreservation of post-thaw culture. We're currently looking in the middle there to integrating the Rotea to our aggregation differentiation steps. However, in the future, we might be able to look to integrating the Rotea to our expansion steps, where the Rotea might have a role in stem cell expansion and cell passaging. It might also be useful for our cryopreservation and post-thaw culture steps, specifically for the cryopreservation harvest and during our post-thaw culture. And with that, some concluding remarks. ViaCyte is developing some stem cell-derived therapies to functionally cure type 1 diabetes. And in order to meet this ambitious goal, we need an ambitious scale-up effort to support our predicted cell therapy demand. The Rotea provides a scalable technology that can support closed and efficient processing of our stem cell aggregate therapy. And our preliminary results show high aggregate viability and normal morphology over consecutive processing days. Thank you for your attention.
Thank you, Prem, Emily, and Eric for your informative presentations. We would now like to start the live Q&A portion of the webinar. If you have a question you would like to ask, please do not hesitate to do so now in your preferred language. Just click on the Ask a Question box located on the far left of your screen. And now let's get started with the questions for BioSite. Our first question is, how can you process larger volumes of around 10 liters with the Rotea?
That is a good question. So as you scale in batch volume, your cell product volume increases as well. Since the maximum hold volume of the Rotea counterflow centrifugation is 10 milliliters of fluidized bed, the path forward would be to process the bioreactor solution in batches, each one comprised of a maximum aggregate volume of 10 milliliters. And this can occur multiple times depending on your load size. And additionally, the high flow kit allows for improved flow rates to support faster processing of these larger batches.
Why did BioSite choose a suspension culture method? In general, what are the overall benefits you see in suspension culture versus 2D adherent methods for stem cell research? Well, perhaps Emily could address this.
Yeah, so the advantage of suspension culture over 2D adherent methods for stem cell production becomes really apparent as the stem cell expansion and differentiation process is scaled. Compared to 2D adherent methods, stem cell production processes utilizing suspension culture combine improved cell processing characteristics such as unlimited capacity for growth, so no surface area limitations, and then at a lower aseptic risk with lower production costs, so reduced equipment footprint and labor requirements.
Thank you, Emily. Our next question. What led to BioSite's decision in selecting the Rotea for bioreactor processing?
So in addition to its ability to maintain aggregate viability, integrity, and morphology, the Rotea's counterflow centrifugation technology is scalable, making it suitable for BioSite's process as we scale up production. Additionally, the Rotea also matches our requirement for repeated batch bioreactor media exchange versus typical perfusion-based processes on the market.
And now this question is for Eric. What are the greatest challenges to Rotea optimization for cell aggregate processing?
Good question. The stem cell aggregates that we work with, they require gentle handling because they can be quite fragile. It's necessary to maintain aggregate integrity during the processing because this is a critical part of their functionality in the final product. So modifying the Rotea process parameters to adequately collect, wash, and refeed the aggregates from the bioreactors, it took some fine-tuning to do, but assistance from Thermo Fisher and working through the parameters to optimize the shear forces was an interesting process. However, we have achieved successful cultures utilizing this system.
And Eric, can you further detail what other processes the Rotea could add value in?
Sure. So as we discussed in the presentation, BioSite has adapted the Rotea to our stem cell aggregate differentiation process. However, we see a multitude of other areas where the Rotea could offer support. One of those includes the final harvest for cryopreservation. When we generate our drug substance, we require an intermediate freezing step for long-term storage, and the Rotea could benefit this process by concentrating, washing, and collecting the final product in cryopreservation media.
Thank you. It looks like we have one final question for BioSite. When adapting your feeding and harvesting process to the Rotea, did you run into issues if the aggregate solution is not uniform? For example, do you observe heterogeneous mix of smaller to larger aggregates?
That is a great question because our aggregates do tend to range in size. We commonly see aggregate solutions that have several standard deviations worth of size, approximately 50% there. But since the aggregates are so much larger than the single cells we want to remove from the solution, we haven't had a whole lot of issues separating the two.
All right, thank you, Emily and Eric. So now that we have finished all the questions that we've consolidated for ViaCyte, we really thank both Emily and Eric for spending the time to deliver this webinar and for evaluating the Rotea system. It looks like they had really good results and application data using Rotea to process pluripotent stem cells. In our next session, we still have some time. I would like to call upon Dr. Premkumar and our panel of technical specialists from Thermo Fisher Korea, Japan, and Taiwan offices to address the broader questions around Rotea or any questions related to cell and gene therapy manufacturing. So feel free to continue to type your questions in your preferred language. We have a number of questions coming in. The Q&A box is located on the far left of your screen. Okay, so on our panel we have Dr. Premkumar, we have Ogita-san from Japan, we have Jasmine from Taiwan, and we have Do-Young Kim from Korea. So let me start with the first question for Prem. What are the steps planned to make it suitable? What are the steps planned to make the Rotea suitable for use in later phase and commercial processes?
Okay, great question. So CTS Rotea System is designed for clinical and commercial cell therapy manufacturing, and the single-use kits are CGMP grade, and both the instrument and the kits are manufactured under ISO 13485. Again, the single-use kits are QC tested individually, and further the kits are gamma sterilized and packed individually. And Rotea's intended use is for research use or manufacturing of cell and gene therapy or tissue-based products. And Rotea software has operator and administrator modes for manufacturing. And to further that, Rotea's 21 CFR Part 11 compliance software solution will also be available in Q3 of this year.
All right, thank you very much, Prem. And now let's move on to the second question, which is, I think, what the audience might want to learn more about. How does the CTS Rotea compare to Clinimax Prodigy from Miltenyi?
Okay, so this is again a very good question. So Rotea is very, very different from the Miltenyi's Prodigy on many fronts. Again, Rotea is an open platform that allows you to use any reagents from any suppliers, and most importantly, it's a modular system. So Rotea combined with an offline incubator would not be tied down over several days or weeks of culture. So this is unlike the Clinimax Prodigy, which does not have an integrated incubator, which makes the scale-up of the manufacturing seamless and grants the user more flexibility to connect Rotea to any culture bags, single-use vessels, or bioreactors upstream or downstream of the process.
Okay, thank you, Prem. And now I'd like to direct you to some questions that came in from Lisa. I think she has two questions. The first, can this system be thought of as a mini bioreactor or fermenter?
Okay, so Rotea is not a mini bioreactor or a fermenter. Rotea is basically a cell processing device, right? So it's specifically intended, as I mentioned, for cell and gene therapy manufacturing. And then there's no temperature control system in Rotea for you to control your cell culture expansion. So it's just a cell processing device. It's a counterflow centrifugation system where you can process your cells upstream or downstream into your bioreactor or cell factories. But again, you cannot use Rotea for expanding any cells. Hope that's clear.
Okay, thank you. And another question from Lisa is, can this system be used to separate two or more cells? I presume she's asking about two or more cell populations with similar physical properties such as cell density, cell size, or diameter.
Okay, so this is a very important question. So as I mentioned, Rotea is a counterflow centrifugation, and because of that unique technology that you can separate cells based on size. So any cells, any cell population with a 3 micron size difference, we could use Rotea to separate. Say, for example, lymphocytes are typically around 6 to 8 microns, and monocytes are around 10 to 12 microns in size. So there's a minimum of 3 micron size that is required for us to separate out lymphocytes and monocyte population. And similarly, it goes like when you want to separate live cells and dead cells. Dead cells are usually smaller in size compared to the live cells. So you can wash away the dead cells using the counterflow centrifugation process, and you can have a very high viable cell in there. And again, cell density plays a major role in the separation. So higher the cell density, the separation efficiency might be different compared to a low cell density buffer. So those are the two key parameters that is required for you to do a cell separation.
Okay, thank you, Prem. I hope that addresses some of Lisa's questions. And now I'm moving on to some questions that were written in other languages. So first, we have a question coming in from Taiwan. So I'll invite Jasmine to read the question, and if you could do a very quick translation in English, that would help the audience.
Okay. So the question is about how involved is the support of for developing a process? Can you help design the program used for our cells? The answer is yes. But since the question is written in Chinese, I will reply in Mandarin as well. 这个问题是说，如果我们要开始做实验，请问你们会如何协助我们？是否可以依照我们的细胞建立实验流程？的确是，Thermo Fisher Scientific 希望支持每一个 Rotea 的客户成功，并且提供客户完整的仪器购买体验。首先，我们会有经验丰富的现场应用专家进行基本的仪器还有软体的介绍，然后在每一个Rotea 的 demo 当中，我们都会针对客户的应用设计适合的 protocol， 然后在客户端用活细胞进行演示，以便客户进行评估。那当客户决定购买仪器之后，我们的FS 跟技术支援团队也会进一步协助客户做 protocol 的开发以及优化。希望这样回答到大家。
Okay, thank you very much, Jasmine. Do you want to give a very quick short statement and respond to that question in English? I think it's about, do we offer development process support?
Yeah, sure. So I repeat the question, whether Thermo Fisher support the customer for developing the process and design the program for their cells. Thermo Fisher Scientific is invested in the success of each Rotea customer and provides support throughout the instrument buying experience. A custom protocol is developed for each Rotea system demo in order to solve a customer application. A live cell demo is performed with the Rotea system for the customer's evaluation. Thermo Fisher Scientific includes initial equipment and software training by our trained field application scientists. Post-sales and FAS and tech support teams were also available for further protocol development assistance. I hope it addressed the question. Thanks.
All right, thank you very much, Jasmine. Now, quickly moving on, we have a question in Korean. Do-young, would you like to read that out and also give a very short response in English as well? Thank you.
Yeah, okay. Now, let me see. Can the Rotea system be connected to multi-layer flask cell as cell factories or cell stacks? Answer is yes. Rotea is extremely flexible. Rotea has been connected to incubators, bioreactors, and cell factories, and G-Rex, and so on. In order to connect it to cell factories, PVC to C-flex tubing adapters will be required. If you are interested in learning more about the technical details making close connection to different vessels, do not hesitate to reach out to us after this call. 안녕하세요. 서머피셔 코리아의 로테아 TS 담당 김정입니다. 질문 주신 부분에 대해서 한국어로 간단히 답변을 드리면요. 일단 로테아 시스템 매우 유연한 시스템이라고 보시면 될 것 같습니다. 다양한 인큐베이터나 생물 반응기, 셀포, 셀팩토리나 G-Rex 등에 충분히 연결될 수 있고요. 만약에 셀팩토리 연결하시고 싶으시면은 PVC C-플렉스 튜브 어댑터가 일단 필요합니다. 다른 튜브에 대해서 폐쇄 연결을 만드는 기술적인 좀 세부 사항에 대해서 좀 더 조언이 필요하시면은 이번 세션 이후에 주저하지 말고 제게 연락 주시면 될 것 같습니다. 감사합니다. Okay, my answer is finished.
All right, thank you, Do-young. And now moving on to a question by Yamamoto-san from the Osaka Uni. So Ogita-san, would you like to take this question?
Okay, can you hear me? Okay,
yes, we can.
Thank you. I'm going to firstly translate the question in Japanese, in English. So the question is, what is the maximum number of cells that can be processed in one time, and how long would it take? So I'm going to firstly answer in Japanese. この度ご質問いただきまして大変にありがとうございます。私、小木田と申します。で、いただいたご質問なんですけれども、ロテアでワンランで処理できる最大の細胞数を教えてください。また、その処理時間はどれくらいかかりますかといただきました。ありがとうございます。結論から申しますと、まず細胞の処理数、これは細胞のサイズに依存しておりまして、T 細胞の場合に 5×10 の9乗個をワンランで処理することができます。で、たとえそれよりも大きな、細胞数になったとしましても、ロテアでループさせて、連続バッチで処理生産することもできますので、例えば、1×10 の10乗個ですと、2ランを連続で処理することをボタン1つで行うことができます。で、細胞のサイズが大きくなりますと、その分処理細胞数が少し減りますが、MSC の細胞等ですと、だいたい 2×10 の9乗個程度でお考えいただければなと思います。で、また処理時間なんですけれども、これは、処理するインプットの処理量に依存しております。で、例えば、1リッターの場合、T セルの処理をしようと思いますと、1リッターあたりだいたい15分から20分ぐらいをお考えいただければ、大丈夫かというふうに思います。はい、ありがとうございます。Okay, I finished answering in Japanese. So I'd like to hand it over to Jacqueline-san.
Okay, thank you, Noble. So we have another question from Mochida-san from Takeda, asking is, so Noble, you could answer this as well. Have you used inline filter in cluster wash concentrate experiments?
I see. Inline filter. Okay. ご質問いただきありがとうございます。インラインのフィルターを、処理経路に、つけて処理した経験があるかといただいたかと思います。 現在ちょっと私、今の持っている情報ではまだ試したことはないんですけれども、原理上は可能だと思いますので、少しその辺の情報を集めた上で、また、ご回答を申し上げたいというふうに思います。Jacqueline-san, yeah, I answered in Japanese.
Sure. Would you like to take that question in English as well? If it's a quick response?
Yeah. Yeah. If possible, could you answer the question if you have an answer?
Sure. So the question in English is, have you used inline filter in cluster wash concentrate experiments? I mean, we can say that it's possible to use inline filters. You could simply filter if it's PVC material to the PVC Rotea kit. And again, the Rotea is very flexible. We don't see any issues at attaching an inline filter. I hope that addresses the questions. Okay. So thank you very much, Ogita-san. And now we have a final question. Do you have any plans to expand the application of Rotea to target more clinical spaces? Perhaps, Prem, would you like to take this question?
Okay, yeah, sure, Jack. Yeah, that's a good question, but we are not going to expand it towards the clinical processing because Rotea is intended only for cell and gene therapy manufacturing, and Rotea is not apheresis processing medical device. Meaning that you cannot use Rotea to perform an apheresis to basically take out from the patient sample by connecting Rotea to the patient by taking out the apheresis. Once you have used a apheresis processing machine to take out a leukopak or an apheresis product, you can again use Rotea to basically process those apheresis material to separate out the PBMCs and then further separate the lymphocytes and monocytes for your cell therapy manufacturing. Hope that answers your question.
All right, thank you very much, Prem. So I think we have addressed all the questions in the chat box. I would like to thank the audience for putting through such interesting questions. So I hope that today's session has been, you've learned a lot from today's session. And again, I would like to thank our three speakers for their time. So in closing, this wraps up our session. We would like to thank BioSite, Emily and Eric for sharing their experiences. That's very valuable to the field. I would like to thank Prem and our technical specialists from Thermo Fisher's regional offices. And a big thank you to you all for joining us today and submitting interesting questions. So if you have any more questions, feel free to send us an email. And we would also like to thank LabRoots for underwriting today's educational webcast. This webcast can be viewed on demand for those who have missed it, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event.